Okay, we're going to discuss all about the non-fermenting gram-negative bacilli. So again, there's still this bacteria here are actually still gram-negative, but they are rod-shaped or spherical because they are bacilli. They are non-fermenting. We're talking about they are not fermenting the glucose, but rather most of them they are oxidizing the glucose. So this group of our bacteria include the following genus. We have your Acetinobacter, Stenotrophomonas, Pseudomonas, and we have your Burkholderia. Okay, we have here the general characteristics of your gram-positive non-fermenting, gram-negative bacilli, non-fermenting gram-negative bacilli. So number one, they grow in your maconchi agar. They're able to grow in the maconchi agar, but they are non-lactose fermenting. So therefore, they appear to be colorless with our maconchi agar. Another one, they fail to acidify with your oxidation fermentation media, especially in the one overlaid with your mineral oil, and therefore they appear to be just able to oxidize our glucose instead of fermenting that one. That will have a change the color only in the open tube, but not in your tube overlaid with your mineral oil. Another one, they fail to acidify your TSIA, and most are oxidase positive. Okay, so we have here again for the maconchi agar, they appear to be, they are able to grow in the maconchi agar, but they appear to be colorless because they are non-lactose fermenter. In the oxidation fermentation, remember that the oxidation fermentation tube, there are two tubes of that. Okay, our positive result for that is yellow na color because the original color of that is green. Okay, there are two tube, one is open, the other one is laid with overlaid with the mineral oil. So they do not able to change the color or they're not able to ferment the glucose on the overlaid with the mineral oil, but only in the one tube na open. And therefore, we consider them here as able to oxidize our glucose instead of fermenting that one. Okay, we have here the triple sugar ion agar and regular ion agar, so they fail to acidify, so they are K over K, meaning to say they are red over red, red on the slant, and red on the path region. Another one, so for the oxidates, so some of them here are oxidase positive, others are oxidase negative. Okay, so oxidase test will identify the enzyme cytochrome oxidase, and the positive result for that would be your deep blue na color. Okay, negative result here, colorless, that would be applicable here with the genus Acinetobacter and your Stenotrophomonas. But in the case of your Pseudomonas and Burkholderia, it will give a positive result that gives here a dark blue na color with the oxidase test. So we start our discussion with Pseudomonas. Okay, so we have here your Pseudomonas aeruginosa. Pseudomonas aeruginosa I consider to be the most clinically significant and prevalent na clinical isolates with this group. Okay, so we have your clinical significance of your Pseudomonas aeruginosa. Pseudomonas aeruginosa account for 75% of your non-fermenting nosocomial infection. So if we talk about the non-fermenting na bacteria causing here the nosocomial infection or hospital-acquired infection, majority of that is being caused here by your Pseudomonas, the 75%. But if we talk about the totality of your nosocomial infection, not only your non-fermenting bacteria, but compared with all the other bacteria, it accounts here for the 5 to 15%. And we regard your Pseudomonas aeruginosa here as the third leading cause of your gram-negative bacteremia. So, pinaka-common gram-negative infection is your Escherichia coli, followed by your Klebsiella pneumoniae. The third one would be your Pseudomonas aeruginosa. So, we have here the significant infections being caused here by your Pseudomonas aeruginosa. The first one, bacteremia. So, bacteremia here in the form of your ecthyma gangrenosum. Ecthyma gangrenosum is more of skin. This is ano siya, infection in the skin, usually on the thoracic area in the lower extremities. Another one is try to cause here your wound infection. Also try to cause here pulmonary disease among our patient with cystic fibrosis. And we have also here also causing the nosocomial urinary tract infection. Okay, and then it also try to cause your other infection like your otitis externa, which are very common among those swimming on the recreational facilities, especially the public facilities like mga swimming pool and all. And also try to cause here your jacuzzi or your bathtub syndrome characterized here by this is a necrotizing skin infection. Another one, it also try to cause your nail infection among those, especially the nail beds of those having artificial nails. And also try to cause here some of your eye infection, keratitis, and endophthalmitis. Okay, then we have here the virulence factor responsible here for the pathogenicity of your Pseudomonas aeruginosa. So the first one, we have your endotoxin. So endotoxin, so this is the toxin that's being attached to the bacterial cells. So basically being gram-negative, so meron siyang endotoxin, which is the LPS component of its outer membrane. Aside from that one, so this is highly motile. So meron siyang flagella, also have here the pili, we have also here the capsule. And it also tries to produce here the exotoxins. So exotoxins, these are being released by the cell here like mga enzymes. You also have here enzymes like your elastase, meron siyang gelatinase, meron siyang lecithinase, meron siyang dinase, and meron siyang hemolysin. The most important na exotoxin produced by this one is your exotoxin A. Okay, the exotoxin A resemble here the toxin produced by your Corynebacterium diphtheriae, which try to block the protein synthesis and therefore try to cause here death and tissue damage of the host tissue. Then we have also here some of the strain of your Pseudomonas aeruginosa, especially recovered from those in the lower respiratory tract of the patient with cystic fibrosis. Try to produce here highly mucoid na colony, and those strain are producing here your alginate. Alginate is the polysaccharide polymer responsible for the highly mucoid na colony characteristic of those strain of your Pseudomonas aeruginosa collected from the lower respiratory tract of the patient with cystic fibrosis. And that strain is also highly resistant to different antimicrobial agents. Okay, we have here the characteristics of our Pseudomonas aeruginosa. So this bacteria is highly pigmented. It tries to produce here a water-soluble pigment that try to fluoresce. So if you try to identify this one with your short na UV light, so this bacteria try to fluoresce because it try to produce this different pigments. The first one, we have your pyoverdine. Pyoverdine is a blue. It's a green na pigment produced by your Pseudomonas. Then we have also here pyocyanine is the blue na pigment. So the combination of that, the pyoverdine and pyocyanine, the blue and the green, is responsible here for the blue-green na color ng ating Pseudomonas with the different culture media, including your Miller-Hinton agar. Okay, then we have also here some of the strain also trying to produce here your pyomelanin, which is your brown na color na pigment. Others try to produce here your pyo rubin, which is the red na color. But the highly specific for the Pseudomonas aeruginosa na pigment is your pyocyanine. Okay, then we have here other colonial morphology characteristics. So we have here, this is strict aerobes. Another one, it try to produce here a characteristic na odor, which is your fruit-like, fruity na odor, grape-like na odor because of the substance 2-aminoacetophenone. Your 2-aminoacetophenone is responsible for the fruit-like odor. Then we have also here, this one would also able to grow at 42 degrees Celsius and I think 35. It can also grow at 35 and 42 degrees Celsius. Okay, this also identified as being positive for the following oxidase positive that will differentiate this one with your Acinetobacter and your Stenotrophomonas. It is also positive for your arginine dihydrolase. Another one is also positive for your citrate. And it also try to produce here your acetamide or try to utilize your acetamide as its carbon source. And the highly selective na culture media for identification for the isolation growth of your Pseudomonas aeruginosa, we have your cetrimide agar. So this is highly selective for Pseudomonas because it contains detergent that will only allow the growth of your Pseudomonas aeruginosa but not your other bacteria. So in our SBA, ship-led agar, it would have beta-hemolytic reaction to clear zone around your colony characteristic, and that would have here characteristic na flat spreading with the characteristic na metallic sheen na characteristic. Okay, the next group, we have your Acinetobacter species. Acinetobacter species here is considered to be ubiquitous because it's one can be found in wide variety of environmental distribution. You can isolate this one in the soil, in the water, and even the foodstuff. Okay, so in adult population, so 25% of the adult population, this bacteria be found in their skin. And the 5%, 7% on the other hand of the adult population, they are having this bacteria on their pharynx. So sa hospital setting naman, you can identify, isolate this bacteria in the following sources. You could have your humidifiers, you could also have your ventilators, you could also have your catheters, or even some other hospital equipments. Okay, this, in terms of the prevalence, causing you the nosocomial infection, this is only second with your Pseudomonas, accounting only for the 1 to 3% of your total nosocomial infections. Okay, then we have here the different species belonging to your Acinetobacter. So this includes here the non-hemolytic species, includes your Acinetobacter baumannii, which is considered to be a saccharolytic. So when speak about saccharolytic, it try to oxidize your glucose. Another non-hemolytic in your SBA would be your Acinetobacter iwaffi. This one is non-hemolytic, just like your baumannii. Only that one do not oxidize nor ferment your glucose. So that will consider here as your asaccharolytic. So the hemolytic strain of your Acinetobacter, we have your Acetinobacter hemolyticus. Okay, we have here the colony morphology with your gram stain results. So this is a gram-negative short plump or cocobacilli. So very short lang siya, cocobacilli or plump na tinatawag. But if you try to isolate this bacteria in your blood culture, it might appear to be gram-positive. Okay, then we have also here colony morphology. So in your Maconchi agar, it appears to be purple na color. And we consider this one as your lactose fermenter. Because by nature, ang mga gram-negative non-fermenting na mga bacteria natin belonging to this group, Pseudomonas, Acinetobacter, Stenotrophomonas, and Burkholderia. So supposedly, di ba, sa general characteristic natin kanina, they are colorless, non-lactofermenter. But in the case of your Acinetobacter, so we consider this one as a lactofermenter because it appears to be purple na color in your Maconchi agar. But in the EMB, using methylene blue, it would appear to be blue-gray with the corn flower blue center on your EMB. Okay, the next genus here, we have your Stenotrophomonas, but most likely the one causing human pathogen, we have your Stenotrophomonas maltophilia. Before, your Stenotrophomonas is actually considered to be plant pathogen, but currently there are some species here which try to cause human infection. So we have here your Stenotrophomonas maltophilia, from the word of maltophilia, so maltose. Okay, just like your Acinetobacter, your Stenotrophomonas maltophilia is also ubiquitous, could be found here on the wide variety of environmental distribution, could be found here in the water sources, could also be found in the sewage system, or even in your plant materials. But in the hospital setting, most likely, this bacteria try to be found contaminating your blood collection system. So, ginagamit natin for blood collection, including your syringe, your mga tourniquet, and all the other equipments. Aside from that one, it also could be found in your disinfectants or even your detergents. And we consider this one as the third leading cause of your nosocomial infection. The third lang siya, number one natin, Pseudomonas, followed by your Acinetobacter. Okay, this bacteria is also primarily intrinsic resistant. So this is already inherently resistant to the following groups of your antibiotics. We have your aminoglycosides, cephalosporin, penicillin, and even your carbapenems. Okay, we have here the colony characteristics. So this is also oxidase negative. Okay, so this is the same with your Acinetobacter. Acinetobacter is also oxidase negative. It's also oxidase negative. A positive lang natin for oxidase would be your Pseudomonas and your Burkholderia. Okay, it's also positive here for the following enzyme, dinase. Also here, catalase. It also tried to hydrolyze your isculene, producing a black colony on that. And we have also here your lysine carboxylase. Okay, in your blood agar plate, this one would have a characteristic na color na lavender green. And in our Makonki agar, this would appear to be bluish in color. So meron din siyang typical na odor. We have your ammonia-like odor. And they try to oxidize dalawang carbohydrate. Number one, they try to oxidize your glucose. And at the same time, also try to oxidize your maltose. Kaya tinawag siyang maltophilia. Okay, then we have here your Burkholderia. So, Burkholderia, just like your Pseudomonas, also oxidase positive. So, oxidase positive by nature ang characteristic ng ating Burkholderia, just like your Pseudomonas. Again, your Acinetobacter and then your Stenotrophomonas are oxidase negative. But this one is oxidase positive. Except this one species later on wherein that one is oxidase negative. But by nature, our Burkholderia are actually oxidase positive. Okay, the first species here of Burkholderia, we have your Burkholderia cepacia. So this species here would eventually be found in the different environmental, okay, or even environmental distribution could be found here in your irrigation fluids or even in your anesthetics, nebulizer, disinfectants, or even your detergents. Porcalia cepacia also associated with those patients. It try to cause here, this is an opportunistic bacteria which try to cause pulmonary infection in the form of your pneumonia among patient with the cystic fibrosis and a patient with the chronic granulomatous disease. So its main manifestation try to cause here rotting of the onion bulb among the plant. It also try to cause a rotting of the foot, nabubulok na okay, body parts in foot here in the part of the human. Okay, we have your colony characteristics. So this one in the BAP appears to be non-wrinkled. Okay, so but this one try to produce here your yellow-green na colony pigment. Non-wrinkled that would identify this one with your Pseudomonas tozeri. Although pareho sila na produce ng Pseudomonas tozeri. Okay, so pareho sila ni Pseudomonas tozeri na produce ng yellow-green na pigment, but yours for Caldera cepacia is non-wrinkled, but this one is wrinkled. Okay, then and the pigment, although try to produce here yellow na green na pigment, but this one do not fluoresce. So, nagpa-fluoresce ang ating pigment produced by Pseudomonas, especially in the short UV light, but this one do not fluoresce. So, the odor for that one is dirt-like odor. And this one is non-lacto fermenter in a makonki agar. Pag meaning to say colorless. However, if the colony here would become old, so like if the culture here would able to have, let's say, 4 to 7 days na, the colony color of this one try to turn here to from the colorless, magiging pink siya. So therefore, parang after 4 to 7 days, it might be interpreted as a lacto fermenter because it become pink in color. But the young colony is colorless, making this one as our non-lacto fermenter. But after 4 to 7 days, it become here pink in color, and we can consider that one already as your lacto fermenter. Okay, we have here selective media for your Burkholderia cepacia. So first one, Pseudomonas cepacia. You could also have oxidative fermentative base, polymyxin, lactose, and bacitracin. Another one, we have your BCSA, which is your Burkholderia cepacia selective agar. So this would really selective na culture media for allowing the growth in isolation of your Burkholderia cepacia. Okay, now we go to the other species of your Burkholderia. We have your Burkholderia pseudomallei. Burkholderia pseudomallei try to cause your malyoidosis. So that one is a pulmonary disease characterized here by acute aggressive granulomatous disease with and try to infect here your pulmonary lungs and even your visceral organ, producing an abscess formation. Okay, so we have here the gram stain result of your Pseudomonas, your Burkholderia pseudomalaise. So in the blood agar plate, it appeared to be a wrinkled na colony characteristics. So again, this would resemble your Pseudomonas tootseri, only that this one is a lacto fermenter. Your Stotzeris do not ferment their lactose. Pero pareho sila na wrinkled na colony. Okay, then we have here your the gram stain result. It would appear to have here bipolar staining. So pag nabing bipolar staining, your safety pin appearance. So mean to say the pinaka-end na part niya may dark stained area and you try to look at the middle portion is light stained area. So bipolar. Dark sa gitna is light stain. Safety pin appearance. Okay, this would have your characteristic na earthy odor, so amoy lupa. Okay, selective media for this one is your ashdown agar. In the ashdown agar, it appears to be deep pink na color, parang siyang red na color. And it's because it tried to absorb the indicator in your ashdown agar, which is your neutral red, kaya nagiging almost red na ang color niya. But it's more of the dark or deep pink na color. And your ashdown agar is being incorporated with the colistin that would only allows the growth of your pseudomalae, but not with the other na species. On the next one, Burkholderia glandulosa. So Burkholderia glandulosa is identified here as being oxidase negative. Because again, ang ating rule dito, lahat sila dapat ay oxidase positive just like your Pseudomonas. With exception here of your Burkholderia glandulosa. So this one is associated with the patient with mga immunocompromised patients, especially having the cystic fibrosis and a chronic granulomatous disease. Okay, especially for those patients having undergone here a lung transplantation. Next, we have your Burkholderia malae. Burkholderia malae try to cause glanders. A glanders is a disease which is a zoonotic infection. So, mean to say, it try to infect our animals, especially for mga livestock like your monkeys, your donkeys, your horses, your mules. This is also a type of pulmonary infections. So it might also infect the human by having that pulmonary infection. So this is identified here as non-motile. Wala siyang characteristic na pigment, wala din siyang characteristic na odor. But delikado ito, so this one has been identified by the US government as one of the agent of your bioterrorism. Okay, then we have here the biochemical reactions for identification of our different species. The first one, we have your nitrate reduction. So nitrate reduction will identify the presence of the enzyme nitrate reductase. So with the enzyme nitrate reductase, it tend to reduce your nitrate to your nitrite. Then you add your sulfonylic acid, will give a pink na color. So positive versus pink, negative is colorless. However, there are some bacteria on which once they try to convert the nitrate to nitrite, they further convert that one to your nitrogen gas, and it might be the reason for having a colorless result. To really discriminate here, that is it really just a true negative ba siya, or it is just the bacteria is just converting that further your nitrite here to your nitrogen gas, then you need to add your zinc dust. The zinc dust here, positive control. So, pag nag-add tayo ng zinc dust, dapat mag-positive na siya. So, like if you add, so if the result here during the first reaction is colorless, okay, then you need to add your zinc dust. Okay, if the result here after addition of zinc dust is pink na color, then we consider that one as a true negative. But for example here, upon addition of zinc dust, it become colorless. Okay, so it would mean here that your bacteria convert your nitrite to your nitrogen gas. That even if you add your zinc dust, it will no longer able to give a positive result kasi ko-convert niya pa din sa nitrogen gas. So, we consider here Pseudomonas and your Stenotrophomonas here as nitrate reductase positive, whereas your Acinetobacter and your Burkholderia as our nitrate reductase negative. They don't have the enzyme nitrate reductase. And then we have also your TSIA, your triple sugar ion agar, even your Kegler ion agar. So we have here the basic reaction with all the others na mga Enterobacteriaceae. Okay, so we have the K over K. The K over K would signify that the slant area is red in color. The butt area is also red in color. So mean to say, do not ferment lactose and sucrose and glucose sa butt area. Wala din siyang gas, wala din siyang H2S production. So again, your gas could be identified here by a crack in your colony, or your culture media, I mean. And your H2S production is identified here by the blackening of the colony. So in this case, wala siyang gas, wala siyang H2S. So we're talking about your Pseudomonas and alkaligenes. Then we have your K over A, that's sa taas, that's your red na color, sa baba is yellow na color, wala siyang gas, walang H2S. That's the case of your Serratia, Shigella, and your Providencia. Then we have K over A, so do not ferment the lactose sucrose sa taas, red, baba is yellow na color, mean to say try to ferment your glucose. Meron siyang gas, saka meron siyang H2S. That's in the case of your Salmonella, your Citrobacter, and we have also your Proteus. Then A over A with gas, walang H2S. Okay, E. coli, Enterobacter, and we have also here your Klebsiella. And we have also here A over A with gas, with the crack in your colony, and H2S with the blackening of the colony. So that's in the case of your Citrobacter frondi. Okay, then we have here your acetamide utilization. So some bacteria try to utilize acetamide as their carbon source, converting that one to carbon dioxide ammonia. Okay, so our indicator for that test would be your brown thermal blue. The positive result is blue. Negative result is green. So this will identify your Pseudomonas aeruginosa. So since, and even your, yan, Pseudomonas aeruginosa. Another one, we have growth here at different temperatures. So growth at 42 and even 35. So that's the case of Pseudomonas aeruginosa and your Stenotrophomonas. But in the case of your Pseudomonas fluorescens, Pseudomonas putida, so they're just only able to grow at 35 but not at 30, but not at 42 degrees Celsius. That will differentiate them from your Pseudomonas aeruginosa because all of them are producing yellow pigment that try to fluoresce. Only that is just differ kanilang growth temperature requirement. Okay, then we have here the summary of our entire biochemical reaction for this non-fermenting gram-negative bacilli. So we have here your Stenotrophomonas multifilia, Cinetobacter ewofi, and your Baumani. So again, these two, Stenotrophomonas and Cinetobacter, are oxidase negative. For the pigment, multifilia try to produce here yellow pigment. Glucose utilization, so they can, they are fermenting. They are oxidizing your glucose. Okay, except your ewofi, which is asaccharolitic. Okay, so they try to oxidize. Oxidize so not ferment. Oxidize the glucose, oxidize the glucose. So oxidize our maltose. Pwede din ating, okay, maltophilia, yes. Your Baumani, pwede siya, but most likely it's more of the maltophilia. Growth at 42. So maltophilia pa din, and we have also your Baumani. Then itong mga sa baba, your Pseudomonas and Burkholderia, we consider them as oxidase positive. So they have deep purple, blue na color. In oxidase test, ito naman colorless sila sa oxidase because they are negative. Okay, then we have here the pigment na pyoverdine. So just applicable lang ito mga pigment natin sa Pseudomonas natin your Burkholderia. But specific for your Pseudomonas aeruginosa is your pyocyanin. Plus, it tried to grow in your citramide agar, acetamide utilization, grow at 42 degrees Celsius. For the differentiation ng dalawa natin, since they are not producing pyocyanin, so you can perform your gelatin hydrolysis test for both your fluorescence and your potida. Pag gelatin hydrolysis will identify the enzyme gelatinase. Okay, ang may gelatinase lang natin would be your fluorescence. Another test here, lysine carboxylase, ornithine, orthonitrophenyl galactopyranoside is for your Cepacia. I think I forgot that one to discuss kanina. ONPG positive ang ating Cepacia. Okay, so that's all with this topic. Okay, thank you.